Yes, Jay sir, you can start.
Thank you very much, Deshmukh sir. Hello, everybody. Good afternoon. Today, in our PhD webinar series, we have our 12th consecutive speaker, Dr. Neha Sharma. Ma'am, I welcome you.
Thank you.
I would like to ask Dr. Deshmukh sir to speak few words.
Thank you very much. Thank you, Jay.
Thank you, sir.
Really, I'm very happy to see the research work of young generation. Young generation in India is a very active, and it is a duty of old people and different organization to encourage, to inspire young generation. We should hear at least what they did during their research. What are the recent advancements in various topics? And I am very happy to tell you that this lecture will be recorded and this lecture will be circulated in all groups of students and teachers. And almost it will reach up to 20,000 microbiologists. So, if only 20 microbiologists hear it, I will consider it is my success. Because the people should learn and people should encourage, inspired by research of Dr. Neha. So I congratulate Dr. Neha for coming on this platform and presenting her lecture. Again, I want to assure Dr. Neha that we are going to give memento and certificate to you.
Thank you, sir. Thank you.
Within 15 days, it will reach to your home address. So that can be a memory for you. Thank you. Thank you, Raj.
Thank you very much, Deshmukh sir. I am grateful that you are inspiring the youth of the country for the upliftment of the subject. Now, I would like to introduce our today's speaker, Dr. Neha Sharma. Dr. Neha Sharma is currently working at Department of Biosciences, Galgotias University, Greater Noida. One thing worth noting is ma'am is currently supervising 2 PhD students. Very nice, ma'am.
Thank you.
Earlier than that, ma'am has worked as Senior Scientific Officer, JITM Diagnostics Private Limited for COVID-19 RT-PCR lab. Earlier than that, she used to work as Research Associate in the project Association and Polymorphism of Lectin Complement Proteins and Expression of Regulatory Protein in Pathogenesis of Rheumatic Heart Patients of Assam at Amity University. Earlier than that, ma'am has worked as a Senior Research Fellow at Amity University. Ma'am has also worked as Junior Research Fellow at the same university and Project Assistant at Corporate Research and Development Center, Bharat Petroleum Corporations Limited, Greater Noida. Dr. Sharma has more than 12 national and international publications in various peer-reviewed journals. Ma'am has participated in more than 15 workshop, training experiences, and conferences. Ma'am, I welcome you to start your lecture.
One more thing, ma'am, please. Dr. Swarnajit Singh is here on our platform. So I also welcome Dr. Swarnajit Singh. He is a senior scientist of India. Thank you.
Welcome, sir. Thank you.
Thank you.
Neha, you can proceed.
Sure, sir. Okay. Thank you, Mr. Jay, for the warm introduction, and a very good afternoon to one and all present in the meeting. I'll be presenting my PhD work, and the title of my PhD work is Association and Polymorphism of Human Leukocyte Antigen class 2 alleles, and lectin complement pathway components in rheumatic heart disease patients of Northeast India. So first, I would like to give a brief introduction about what is rheumatic heart disease and how it develops. The causative agent is group A Streptococcus pyogenes, which is most common bacterial cause of pharyngitis, and it is responsible for approximately 15 to 30% of cases. So pharyngitis is basically characterized by sore throat, scratchiness, and pain in the throat. And if left untreated, it may trigger rheumatic fever in genetically predisposed children. That is 0.2 to 3% of the total cases. Further, it leads to rheumatic fever. According to Jones criteria, which has recently been revised by American Heart Association, the disease manifests as carditis, polyarthritis, chorea, erythema marginatum, and or subcutaneous nodule. Out of all the manifestations, carditis is the most serious complication of RF. It occurs in more than 60% of RF patients and leads to rheumatic heart disease. Now, coming to the rheumatic heart disease, it is majorly characterized by thickening of aortic and mitral walls. The early diagnosis of RHD can be done through echocardiography, which is characterized by a prolonged PR interval that is due to the thickening of the walls and the backflow of the blood. And the pathogenesis of the rheumatic heart disease, the major attributable factors are susceptible host, aberrant immune response, Streptococcus pyogenes. Apart from these factors, social and environmental factors also impart differences in risk in different populations around the world. Out of all these factors, in this study, we majorly focused on susceptible host and aberrant immune response. Now, the current status of RHD in endemic and non-endemic countries. So this is the distribution throughout the world. And the global estimated death rate is approximately 2 lakh to 3 lakh. Most deaths occur in low and middle income countries. 33 million prevalent cases of RHD are there worldwide, and more than 9 DALIs are reported throughout the world. If we specifically look at the distribution of RHD in India, so it is strikingly variable. And if you look at the Northeast Indian population, it is most prevalent in Assam, followed by Meghalaya and Nagaland. Now, as I said, in this study, we have majorly focused on the genetic predisposition and the aberrant immune response. So I'll discuss it one by one. If you look at the genetic predisposition, so there are studies of families and twins. They have shown the significant correlation between these two and emphasized its role of genetic predisposition in RHD. In our study, we have focused on human leukocyte class 2 alleles. And apart from that, we are focused on lectin complement pathway components. 3 proteins of this pathway, MBL, 5:2, and MASP2. So I'll just brief you about the current literature of the association of HLA class 2 alleles and RHD. So worldwide, many studies have reported association of HLA class 2 alleles, and there is a pattern observed. HLA-DR7 has been reported in maximum of the populations. But interestingly, DR7 has never been reported in Indian population. And in Indian population, few studies have reported this association from North India and South India, but there is no data available from Northeast India. Now, the second aspect is the lectin pathway in RHD. If you look at the left side of the figure, so these 2 proteins, 5:2 and MBL2. So lectin pathway is one of the pathways of the complement system, which activates the pathogen clearance after binding of 5:2 or MBL2 on with the proteins present on the surface of the microorganisms. After that, it complexes with MASP1 and MASP2. These are mannose-serine associated proteases, which leads to the catalytic activity of other complement proteins and hence lead to the complement activation and inflammation. But in case of rheumatic heart disease, the same process will be taking place inside the heart walls, which leads to the inflammation of the walls and thickening of the walls. So due to this, so if there is any alteration or genetic modification in these proteins, that will lead to the less clearance of the pathogen, and that is how it is associated with the pathogenesis and the increased susceptibility towards RHD. So there are few studies which have reported association of lectin pathway components in RHD. MBL2, elevated serum levels have also associated, and low MBL serum have also been reported, and they are linked to the SNPs present in the promoter region and the exon 1 region of the MBL2 gene. Whereas in phycolin 2, low serum levels have been reported and they are associated with the SNPs in the promoter region. Whereas for MASP2, low serum levels have reported and are associated with certain haplotypes present in intron 9 and exon 12. This is the global scenario. If we look at the Indian scenario, only 2 studies are there. First study, which is the study is being done by my supervisor only. So they they analyzed actually the exon 3 of MBL2 gene, but no significant correlation was found in North Indian population. Another study was reported from South Indian population, and they found a novel mutation in the exon 1 of MBL2 gene. So overall, taken together, we can say these studies confirm the significant association of HLA class 2 alleles and lectin pathway components with rheumatic heart disease worldwide. But there was no data which was available for Northeast region of India. And moreover, the incredible ethnic diversity of Northeast India emphasized the potential role of host genetic factors in the pathogenesis of RHD. So the aim of my study was to investigate and clarify the role of these molecules in susceptibility and progression of RHD. I'll just give you a brief about the methodology. So we enrolled the patients from Assam Medical College and Hospital, and the sample size was 100, and few inclusion exclusion criteria were set. So clinically diagnosed RHD patients, which were confirmed by echocardiography, they were included, and along with the Northeast ethnic originicity. Then the exclusion criteria, so patients were not from the same ethnic origin, they were excluded, and patients with the history of valvular heart surgery were also excluded from the study. So for the SNP studies, first of all, we did the HLA, SSP, PCR based HLA typing, and then HLA allele was amplified, and the amplified product was sent for sequencing. Then bioinformatic analysis was performed using BLAST, MDR, PROVEAN, and homology modeling that I will be explaining later. For serum level analysis, ELISA was used. And for SNP analysis, MBL2 gene, phycolin 2, and MASP2 amplification was done, and the amplified product was sequenced. Now, I have divided the major findings of my PhD work into 4 sections to make it more understandable and clear. So I will discuss each section one by one. So section 1 is clinical and socioeconomic profile. This table shows the demographic details of the patients enrolled for the study. Number of female patients was slightly higher as compared to males, and ethnicity, major ethnic groups were Ahoms, T tribes, Assamese, Kachri, Brahmin, Kalita, and there was a mixed population as well. Now, if we look at the various manifestations of rheumatic fever, so majority of the patients reported carditis as expected, because it is the most complicated and severe manifestation of the rheumatic fever. Then the patients were distributed based on their severity. So mitral stenosis, mitral regurgitation, and mitral aortic regurgitation. And the percentage shows the number of severe cases in each category. Now this shows the various symptoms of rheumatic heart disease, and majority of the patients shows palpitation and easy fatigability, which are the true clinical representation of the carditis. Apart from clinical characteristics, we focused on the socioeconomic parameters of RHD as well. So we took various parameters which actually represent the socioeconomic status of a population, like dwelling location, housing type, cooking fuel, monthly income, and education. And using logistic regression multivariate analysis, we observed that housing type and monthly income and education were the significant factors. Although worldwide, overcrowding is considered to be the most significant factor, but in our study, we did not find any significance. That might be due to, it means it depends on the number of people staying in a room or in a house, but that also depends on the size of the room, and there are other factors. So based on that calculation, we could not find any significant association of overcrowding with this population. Rather than that, housing type, monthly income and education were found to be significantly associated. Later on, we published this data in European Journal of Preventive Cardiology to show the association between the socioeconomic status and to also depict the clinical spectrum of rheumatic heart disease patients. Later, my supervisor suggested that why don't we have a look over the worldwide representation of these factors. So I just, it was a kind of a systematic review. So I went through all the studies I came across, and we looked for the factors which can be significantly, which can significantly impart risk towards the increased susceptibility. And we compiled this data and we published it in Current Infectious Disease Reports in 2019. Now coming to the section 2 of the major findings. So HLA class 2 alleles. We did the SSP PCR based HLA typing, and among the HLA DQB1 alleles, HLA DQB0301 was found to be the most predominant and significant one. Then we checked the distribution of this allele among the various ethnic groups, and it was observed that this allele was positive and it was present in majority of the patients belonging to the home group. So that is how we compared the presence of this allele in different ethnic groups. Later, we checked for the distribution of this allele on the basis of severity as well. And it was observed that the majority of the severe cases of mitral stenosis, mitral regurgitation, aortic regurgitation, they were positive for this allele. And moreover, mitral wall thickening and aortic wall thickening patients were also positive. Majority of them were also positive for this allele. It was also associated with the rural population because it was more predominant in the rural population. So overall, from this data, we can conclude that HLA-DQB103 can be a significant susceptibility marker in this population. And it is also predominant in rural population, whereas it is also associated with severity of the disease. Then we focused on the another class. We published this data in Asian Cardiovascular and Thoracic Annals in 2020 only. Then we focused on another class, which is HLA-DRB1. So among that, HLA-DRB1501 was found to be the predominant allele. And similarly, as for HLA-DQB1, it was also predominant in most of the severe cases, and there was a difference in the pattern. In HLA-DQB1, it was predominant in both aortic and mitral wall, whereas HLA-DRB1 was specifically predominant in the mitral wall thickening cases. So that's how it is related to the severity of the patient as compared to controls. Then to understand what was the difference, what was the variation that was making them predisposed towards the disease, we amplified the PCR, this particular allele, which was predominant, HLA-DRB1 allele. And after amplification, sequences were aligned for patients, controls, and the HLA-DRB15 sequence was taken from IMGT HLA database. After alignment, we observed 3 SNPs. And the genotype distribution of the 3 SNPs was checked. So in SNP1, GG genotype and the T allele was found to be significantly associated. In SNP2, TT genotype and the G allele was found to be significantly associated, whereas no correlation was observed in SNP3. So overall, we can say in HLA-DQB103 and HLA-DRB115, DQB1, we only checked the distribution, but for DRB15, we found significant SNPs which were present in patients and can be one of the factors for the increased susceptibility. Now coming to the third section. Now, as we have observed that there was variation in the sequences, but what is the impact of this variation? So we performed in silico analysis to understand the effect of these SNPs on structure and function of MHC class 2. So first of all, we used protein variation effect analyzer. So we translated the sequences of the patient's controls and the HLA-15 into the protein sequence and aligned them. After alignment, we observed 5 major variations. Out of those 5 major variations, 2 of the variations were deleterious, and out of those 2, only 1 was statistically significant. And one of the first one, that is D70H, aspartate and histidine, it was deleterious and it was present in the binding region of the MHC class 2. Whereas the last one, T119T, which was also deleterious and significant, it was present in the non-binding. Although the binding one is not significant, but we analyzed both because the impact of the variation which is present in the binding region will be more as compared to the non-binding region. So we modeled the structure of HLA-DRB15 and observed that two of the variations are marked in red color and they were present in binding and non-binding. This, the one which is in circle, it is the non-binding region. And in this, proline was replaced by threonine was replaced by proline. So based on the stereochemistry of these amino acid proteins, since proline is not a very simple linear structure, that replacement will impact the interaction of amino acids present in that region and ultimately will impact the structure. So that structural difference in MHC will, since it is not in the binding region, but still it might impact the structure in the cytoplasmic tail. Then we analyzed the amino acid substitution in the binding region, because if it is in binding region, it is directly going to impact the peptide binding and the presentation of the peptide to the T cells and ultimately the activation of the T cells. So in this case, aspartate and histidine replacement was that. Again, the problem was whenever we switch this amino acid, so it is going to be alter the structure of the MHC and change the groove, binding groove of the MHC class 2. Now, so that's how HLA class 2 was associated with RHD. Now, this section 4, and that is the last section, lectin complement pathway and RHD. So we focused on three major proteins. First is MBL2 gene. MBL2 gene polymorphism. We amplified the MBL2 gene exon 1 and we observed that 3 SNPs were there. Out of that, one, the RS number is already mentioned in the table. It is already reported to be associated with MBL2 serum levels and its activity. So we also find this significant genotype in our population. And we observed that GG genotype and the dominant model were associated, significantly associated in patients. And we also performed the HW analysis and all the results were consistent with the Hardy-Weinberg equilibrium. Similarly, we performed the amplification of the 5-2 gene for the promoter region. In the promoter region, it has already documented that if there is any variation, it is going to impact the serum levels as well as the activity and the binding capacity of the 5-2. In our study, we found one of the SNP of the promoter region to be significantly associated with RHD patients. We also looked at the haplotype profile, but in haplotype profile, there was no association found. The numbers were not significant. And similarly for MASP2 also, no significant correlation was found between SNP and low serum level of MASP2 with increased risk towards RHD. We also analyzed the serum levels. So if MBL2, 5-2 and MASP2, these were the three major complement proteins we analyzed. And as we can see, for MBL2 and 5-2, there is a significant difference in patient and controls, and for MASP2 as well. But the SNP we analyzed in our study was not associated with the serum level of the MASP2. So definitely there are other SNPs. We did not analyze that. But the one which could have been associated, we predicted that one. But in our study, there was no significant relation between these two, although MASP2's serum levels were low as compared to controls. And similarly for MBL2 and 5-2, but in 5-2, we have found the correlation with the promoter polymorphism. And for MBL2, we have found the association with the already reported genotype. Now, the fourth protein, which I did not mention earlier, so MAP44. MAP44 is a protein which acts as a competitive inhibitor of lectin pathway activation, and it can displace MASP2 from MBL. So it hence prevents the cleavage of C4 and C2. So that is how it can lead to the inactivation of the inapt functioning of the complement system, and ultimately the less clearance of the pathogen. So this is how the lectin complement pathway components are associated with RHD. Now coming, and this data we have recently published in Human Gene, which shows the lectin complement pathway component as risk factors for rheumatic heart disease in Assam, India. So coming to the summary and conclusion, the major findings are socio-economic factors such as education, dwelling location, monthly income, they have been found to be significantly associated with increased risk of RHD in this population. Genetic predisposition study, HLA DQB103, HLA DRB115, they were the predominant alleles in RHD patients of Assam, and both alleles were predominant in rural population as well as in the severe cases of mitral regurgitation, aortic regurgitation, and mitral stenosis. So SNP in HLA DRB115 allele, T119P, which was the in silico analysis, showed that this substitution was present in the non-binding region of the allele and imparts a deleterious effect on the structure and function of MHC. And although it was not significant, but we also observed one variation in the binding region as well. And then the lectin complement pathway proteins in RHD. So single amino acid substitution at one of the primary promoter positions imposes an increased risk toward RHD, and low serum level of MASP2 against the high level serum of MAP44 suggests inapt functioning of the lectin complement pathway in RHD. So that is how we studied these parameters and tried to find the positive correlation with increased risk so that we can screen the predisposed population and we can have the early intervention. So I would like to thank Microbiology Society India for providing this opportunity to present my work. Professor Narayan Rishi, who is advisor, Amity Institute of Virology and Immunology, Amity University, Noida. My PhD supervisor, Dr. Devinder Tur. My co-supervisor, Dr. Lokajit Baro. He's from Assam Medical College and Hospital. And my current affiliation, Galgotia University, Greater Noida. I would also like to thank the funding agencies, ICMR and DBT. Thank you so much.
Thank you very much, ma'am. Oh, it was an excellent presentation.
Thank you. Thank you so much.
Now we are open for question and answer session. So anybody who might have questions, please unmute yourself and you can ask.
I have one question.
Yes, sir.
Jay,
Please, sir. Please.
can I ask?
Sure, sir. Please.
Why did you choose Northeastern India for this study? Why did you choose Northeast India for this study?
Yes, sir. So first of all, when we looked at the data of RHD from Indian population, there was no data available from Northeast India. And moreover, the ethnicity, the diversity in the ethnic groups of Northeast India motivated us to choose Northeast Indian population.
So you took mostly from Guwahati, Assam?
No, sir. We took samples from Assam Medical College and Hospital. So people who are coming from Meghalaya or maybe any other state of the Northeast India, they are also included. So it was not specific to that.
That makes sense, because you should have from the whole Northeast, like Nagaland,
Yes.
Mizoram, Manipur, right?
Yes, sir.
That's very good. That's a very good study. Now, you have got 4 proteins, right?
Yes, sir.
What is the difference in amino acid composition of these 4 proteins?
After analysis, you're asking?
Yes, yes. What are the differences in these 4 proteins?
Okay, so in these 4 proteins, we analyzed only for 3. The last protein was checked because it is an inhibitor of complement system. We did not analyze the amino acid sequence. For other 3 proteins, we observed that phycolin 2 promoter region, there was a change of threonine and proline. Okay. And in HLA, there was a change. Actually, not phycolin. For HLA, there was a change of threonine and proline, aspartate and histidine, whereas there was polymorphism in phycolin 2 and MBL2.
I see. So the amino acid was different in all the proteins, 3 proteins? Amino acid composition.
Amino acid composition in all the 3 proteins? No, we did not check. The reason was because in phycolin, it was in the promoter region. So definitely it is going to impact the serum expression, right? And in MBL2 and MAS2, we did not find the correlation. That is why we did not check.
Why I was saying, if you can compare the amino acid composition
Sure,
of these
sure.
3 or 4 proteins.
Your voice is
Give
breaking, sir.
you
Excuse me,
a
sir.
very good publication,
Thank you, sir. Thank you.
because even if, yes,
So,
Okay,
sorry,
thank
please
you.
continue. Your voice was breaking down, but now you are audible. Sorry. Please.
Now it's okay. Now it's okay.
Yes, yes, you're
All right.
audible.
So what I was trying to say that if you compare the amino acid composition of these 3 or 4 proteins,
Yes,
this will lead to a very good publication.
sure, sir, sure. We'll
Because
consider that.
even one amino acid difference makes a lot of difference
Yes,
in the
yes,
protein
definitely.
composition
Definitely.
and activity, right?
Definitely.
And you can
Thank
also
you for the
test.
suggestion, sir. Thank
You
you.
can also test the thermostability of this protein.
Okay.
Some proteins may be thermostable.
Yes, sir.
So, just heat at 45 degrees centigrade
Yes, yes, yes.
and report the protein if it is thermostable.
Yes, sir.
Thank you so much. Thank you.
Thank you. I have noted down your suggestions and will
Very
definitely consider that.
Very
Thank
good.
you.
Okay, Jay, you can
Thank
ask
you, sir.
something. Thank you.
Professor Basavaraj want to ask.
Yes, right, sir. Yeah, Dr. Neha, it is a very exciting research, at least for me.
Thank you, sir.
Yeah, I would like to know. You have, mentioned that the living conditions and other social
Economic.
components are going to play.
Yes.
They are like predisposing elements.
Hmm.
How would you, scientifically correlate that one? Second component is, yes, your study with respect to lectin pathways and so on.
Okay.
Could able to correlate to the pathogenesis.
Yes.
I would like to know whether your investigation will guide us in case of any therapeutic application or thought process.
Okay, sir. So, sir, first part of your question was about socioeconomic status. So it has been seen that RHD is very much prevalent in the low socioeconomic populations, first of all. The reason might be because of the increased exposure towards gas infection. Because people who are living in low sanitation conditions and overcrowding conditions, they are more prone towards the infection. That is the one basis. But overcrowding cannot be the only factor which represents the socioeconomic status. So that is why we need to take all the parameters which truly represent the socioeconomic status of a population. So I hope it's clear now.
Yeah, scientifically, how we correlate? For example, maybe it is nutritional element or
Nutritional, definitely nutritional element is also there, but the major focus which has already been documented is the exposure to gas infection. So people are living in the overcrowding areas, they are more exposed towards the infection, and they are not even aware about this because of the education, I'll say. Is it? I hope it's clear, sir.
Yeah. But I think we need more investigation, maybe not by your team, maybe, or you can look into future aspect of correlating these things.
I will explore,
fact,
sir. Maybe I might be missing some point. I will also explore.
It's not straightforward, but maybe this data will be
Yes, it is not a direct link. That is true, sir. It is not a direct link. That is true. Yes, sir.
Any therapeutic interventions from your study? That is my second.
Yes, sir. Since I have found only this thing, the expression of the phycolin-2 serum low level were there, and it was associated with the promoter activity. So that can be targeted for the therapeutic purpose. Other than that, MBL and MASP2, they were not significant, although low serum levels were there. So we might supplement them as a therapeutics. Other than that, we did not find any correlation.
Thank you, sir. Thank you, ma'am. Do we have any more questions from anybody?
Sir, I will continue. I will just for
Please.
another clarification.
So,
is there any inheritance among the generations?
Yes, sir. There are
with predisposing. Yeah, we have a family history of my sister had this rheumatoid fever in the young age. Unfortunately, she has to undergo the wall, treatment in the elder stage at the later stage of life. For the next generation, what could be the preventive? This one we can think, or
Yes, sir. So genetic inheritance is there definitely. And to protect the next generation, we need to have the early diagnosis and intervention.
yeah,
So if
you mentioned some diagnostic component.
exactly. So penicillin treatment should be continued. In this case, primary prevention and the secondary prevention is the penicillin treatment, which should not be discontinued. And if a person is infected with the same, means if there is a recurrence of the infection, that should be treated immediately. Otherwise, it will lead to the progression of the disease. So we can have early screening, early diagnosis, even if there is not a single symptom of the heart failure, sorry, not heart failure, no, the heart disease symptoms are not there. Even in that case, we need to follow up with the Jones criteria. If that, I hope I talked about Jones criteria. So Jones criteria is about the manifestation of the disease. So if there are two major and one minor manifestation, or at least two minor and one major manifestation, then person should undergo echocardiographic analysis. So that is how we can have the early diagnosis, early intervention, and we can prevent the disease in the next generation.
Okay, thank you.
Okay. Thank you, sir.
Thank you, sir. Thank you, ma'am. Dr. Sharma, I would suggest you please type your email address in the chat box. So if any further questions, they can direct it to you.
Sure.
Nice brainstorming session, everybody. And like Dr. Deshmukh sir has said, you will receive the memento at your home in 15 days.
Thank you so much. I have shared my email ID.
All right, ma'am. Thank you very much. Thank you, everybody. Have a nice weekend.
Thank you so much, Mr. Jay, for coordinating everything so well. And thank you, Deshmukh sir, for providing me this opportunity. It's definitely a wonderful platform to share our work. So thank you so much.
Thank you, Deshmukh sir. Thank you, everybody, for being here. Thank you, ma'am.
Thank you. Thank you, organizers, for initiating.